always a good start to give away prizes. Without further ado, let's go to the next speaker. And it's a pleasure for me to make way on stage for the youth. And I think it's very important as an international society and a scientific society that we also are a stimulating environment for young people who are involved in research and are getting enthusiastic on anesthetic pharmacology. So the next speaker is one of our PhD candidates who is working on interactions, and she happens to be very handy with computers, was one of the interested trainees in the non-MEM course of Steve Schaefer, and I have to admit, she's much better in it than I am, or ever will be. So we've done some work on a new index, the Noxious Stimulation Response Index, and she will present this index as I see it is already used on some posters, so might be interesting to get a good view on what it is, where it comes from, and what the capabilities will be for now and the future. So Laura Hannivoort, welcome on stage.
All right, thank you, Hugo, for the kind introduction, and I'm honored to be invited here to honored to be invited to be presenting our research here at this meeting. And today I will talk to you about the Noxious Stimulation Response Index, which is a calculated parameter which we base on our novel 3-drug interaction model and its possible uses for better drug titration in clinical practice. So first of all, I'd like to declare a conflict of interest, as our Department of Anesthesiology has received non-restrictive educational grants from Draeger in Lübeck, Germany. And with that out of the way, I'd like to start with something we likely all know about, the concept of MAC. And MAC is, of course, the minimum alveolar concentration at which 50% of patients will move during a surgical stimulus or skin incision. And MAC, as you all know, is not a measured parameter, but it's a calculated parameter derived from population studies, and it's used daily in clinical anesthetic practices. However, there are several downsides to the MAC. One of them is it's based only on one drug, the volatile anesthetic. Whereas in clinical practice, the volatile anesthetic is rarely the only drug that will influence whether a patient moves during a noxious stimulus or not. And often opioids are added, and propofol induction may be used before sevoflurane is started or before volatile anesthetic is started. But also drugs such as midazolam, ketamine, clonidine may be added during anesthesia, which will influence the probability of tolerance of a noxious stimulus. And the MAC does not take this into account, although a clinician will keep this in mind, of course, and extrapolate from the MAC. But this will still be with a risk of either overdosing or underdosing a patient during anesthesia. And of course, patients are being anesthetized without the use of volatile anesthetics, and the MAC is altogether useless in those cases. So the goal was to improve on a concept which is already often used in clinical practice and make it more applicable to the different combinations of anesthetics that are used in daily practice with or without the use of volatile anesthetics. For instance, with intravenous sedatives and opioids, but also including the interaction between 2 or even more drugs. So why exactly is this necessary, apart from the downsides of MAC I already mentioned? Well, there are several so-called depth of anesthesia monitors available, but these tend to correlate mostly with the hypnotic component of anesthesia and not so much with the balance between nociception and anti-nociception, nor with immobility. So the goal of so these monitors are usually relatively poor predictors of whether a patient will tolerate a noxious stimulus or not, and therefore the last few years the challenge has become to find a parameter which will accurately predict tolerance of a noxious stimulus. So with this idea, Schumacher et al. First presented the Noxious Stimulation Response Index 9 years ago at the ISAP meeting of 2007, which was based on the interaction study by Bouillon in 2004 between propofol and remifentanil. And 3 years later, Martin Lukenbühl published the NSRI in Anesthesiology. based on the interaction study by Struijs et al. In 2003, also based on propofol and remifentanil and interaction. So first let me explain what the NSRI is. The NSRI is a calculated parameter on a scale of 100 to 10, where at 100 the patient has received no anesthetic drugs and should therefore be awake. And at near 0 there's a profound drug effect and the patient should be very deeply anesthetized. The NSRI is actually a transformation from the probability of tolerance of laryngoscopy. So in this case, it means that the patient shows no motor response to laryngoscopy, which is a noxious stimulus. And in fact, Thomas Bouillon already coined the PTOL as an alternative to MAC back in 2004. So the PTOL is transformed in such a way to the NSRI that by definition, PTOL of 0.5 corresponds with an NSRI of 50, and a PTOL of 0.9 corresponds with an NSRI of 20, which means that at an NSRI of 20, only 10% of patients will respond to by moving to laryngoscopy. And for the calculation of PTOL, the parameter U is used. And U is the combined potency of the used drugs normalized to their CE50, or the effect site concentration, which produces 50% of the maximal effect of each drug. And the formula for U shown here is only an example of how the combined potency of a drug, of 2 or more drugs, can be calculated. And I shall explain more about that a bit later. And the gamma is the slope parameter of the formula, or the steepness of the concentration effect relationship. As I said before, there are several methods to calculate U, the combined potency, and as there are several different interaction models. And I'll only focus on the interaction models which are relevant to our study. The simplest interaction model is the additive model, in which the normalized concentrations of each drug are added to provide U. The Greco model is a little bit more complicated by adding an interaction parameter alpha. And if the alpha is 0, the interaction is additive, and you will reach the same formula as the additive model shown above. And if alpha is smaller than 0, the interaction will be infra-additive. And if alpha is greater than 0, the interaction will be supra-additive or synergistic. The reduced model assumes alpha to be 1, which is by definition synergistic, and also removes the additive component of drug B, resulting in the formula shown here. In this formula, CE50 of drug B is the concentration that apparently reduces the CE50 of drug A by 50%, or strengthens the value of U by 100%. The hierarchical model is more complex, and when applied to anesthesia, it describes an interaction in which a stimulus with a certain pre-opioid intensity is first attenuated by an opioid drug, and the higher the concentration of the opioid, the more the attenuation of the intensity of the stimulus is. And this post-opioid intensity will then go through the effects of the hypnotic drug and resulting in a certain probability of response or probability of no response. And the formula has been simplified because not all the original parameters can be derived independently of the original model. And in this model, only the post-opioid intensity remains to be estimated. So when only a single stimulus is investigated, the intensity of the stimulus can be set to 1, which results in the formula shown here, which is actually very similar to the reduced Greco model with the addition of a slope factor for drug B, or in our case, the opioid. So there have been several interaction studies between remifentanil and volatile anesthetics in the past, but the endpoints for these studies were usually the reaction to a surgical incision, also known as MAC reduction studies, whereas the NSRI is based on tolerance of laryngoscopy. So new studies were needed to expand the NSRI, which already exists between propofol and remifentanil, to include sevoflurane. And as sevoflurane is also often combined with the propofol induction bolus, sevoflurane-propofol interaction studies were also necessary on top of the sevoflurane-remifentanil interaction study. So in the past years, 3 large 2-drug interaction studies were performed, which were similar in study design, namely the crisscross design, and also with similar study endpoints, with usually a series of stimuli, but all of the studies including tolerance of laryngoscopy, which is of course important for the NSRI. And for all studies, the use of intravenous drugs was, the administration of intravenous drugs was using step-up target controlled infusion, with also an equilibration time of 12 to 15 minutes to ensure equilibration between plasma and effect site concentrations. So the first study I already mentioned before, the propofol-remifentanil interaction study by Bouillon. The investigators found a synergistic interaction, best described by the modified hierarchical model. And as only one stimulus is relevant for our study, namely tolerance of laryngoscopy, the stimulus intensity can be set to 1 and eliminated from the formula. And Schumacher et al. Investigated the interaction between sevoflurane and propofol in 2009, and they found an additive interaction, which is the same as the Greco model, with an interaction parameter alpha being 0. And Heise et al. Investigated the interaction between sevoflurane and remifentanil, and like for propofol and remifentanil, they found a synergistic interaction best described by the modified hierarchical model. So our purpose was to combine these 3 interaction models to one triple interaction model. And since both sevoflurane-remifentanil and propofol-remifentanil interactions follow the modified hierarchical model, we presumed the same to be true for the triple interaction model, with an additive interaction between sevoflurane and propofol. And in the original 3 studies, different values were found for a CE50 for remifentanil, for the gamma for the opioid, and also the gamma for calculation of the PTOL between sevoflurane and propofol as they interact with remifentanil. So our hypothesis was that these values would be different in the eventual model as well, where each parameter could best be described as a combination of the parameter for sevoflurane and for propofol, depending on the fraction of sevoflurane used in the total propofol-sevoflurane mix. So in our pooled analysis, we found that the CE50 for remifentanil and the gamma do not differ between sevoflurane and propofol. And we also found that the gamma for the opioid is not different from 1. And as I explained before, the modified hierarchical model is the same as the reduced Greco model with the addition of a gamma for the opioid. And as this gamma for the opioid is equal to 1, the final model is actually a reduced Greco model with an additive interaction between sevoflurane and propofol. So those of you familiar with 2-drug interaction models might be expecting a nice 3-dimensional response surface model, but I'm simply not clever enough to create 4-dimensional response surface models on a 2-dimensional screen. So we're going to have to do with separate 2D graphs of isoballs. And somewhere I think physics may also play a role in me not being able to do that. So this slide shows 3 graphs for the different 2-drug combinations: propofol-remifentanil, propofol-sevoflurane, and sevoflurane-remifentanil. But for readability, I'll focus on only 2 panels. Panel B is sevoflurane-propofol, and panel C is sevoflurane-remifentanil. And the green lines you see here are the PTOL 90%, the PTOL 50%. The blue squares are the non-responders, or the patients that tolerated laryngoscopy, and the orange squares are the patients that responded to laryngoscopy by moving. And in this graph you see the PTOL 90 here and the PTOL 50 here. And what you can see is that for sevoflurane and propofol there's an additive interaction as indicated by the straight line, whereas in sevoflurane-remifentanil the line is curved inwards. Also on this graph you can see the BIS 40, the bispectral index of 40, and a bispectral index of 60 as lines as well. And this is considered as an adequate range of anesthesia. And here is the BIS 40 and the BIS 60. And there are several things that are that stand out. One of them is that there's a large overlap between non-responders and responders in the sevoflurane-propofol interaction, and also that almost all of the non-responders, all those who tolerated laryngoscopy, have a BIS below 40. So with only sevoflurane and propofol, a BIS of 40 will probably still not be enough to ensure a tolerance of laryngoscopy. Actually, as you look here, the PTOL 50 is far above a BIS 40 line. So even at a BIS of 40, between these two drugs, you're going to have more than 50% of people who will not tolerate laryngoscopy. And this is likely attributable to the absent or minimal analgesic effect of both sevoflurane and propofol. Also that both drugs significantly affect EEG parameters such as the BIS, but not as much tolerance of laryngoscopy. In panel C, what stands out is that at low opioid doses, the PTOL 90 moves far above BIS of 40 again, like same as here. So only at a concentration of remifentanil of lower than 1.5 nanograms per milliliter, you need a BIS of at least 40 to ensure a good probability of tolerance of laryngoscopy. On the other edge of the spectrum, the PTOL 90 falls below the BIS 60. So at higher concentrations, starting from a little under 4 nanograms per milliliter, BIS of 60 will be plenty to ensure a good probability of tolerance. And this is likely due to the fact that remifentanil has a profound effect on tolerance of noxious stimuli, but only has little effect on EEG parameters. So one of the possible applications for this model and the NSRI is that if one wants to use a sevoflurane-remifentanil anesthesia with propofol induction, you can calculate the time at which to start you start your sevoflurane. And the above panels show for different propofol boluses and different remifentanil concentrations when to start your sevoflurane to ensure a probability of tolerance of laryngoscopy of 90%, which is by definition an NSRI of 20. And again, for readability, I'll show only 2 examples. And in this graph, the effect site concentration of sevoflurane is green, propofol is black, remifentanil is red, and the PTOL is the blue line. And at low doses of remifentanil and propofol bolus, sevoflurane has to be initiated early and dosed to a high concentration. You have to start it at around 200 seconds, whereas at higher doses of propofol and higher remifentanil concentrations, you can start it at around 400 and, of course, dose a lot lower. And this is logical to any one of us, but the NSRI can actually help us determine when and how high to start your sevoflurane. And to do a primary validation of the NSRI, we calculated the prediction probability, the PK. And for this, we divided the data set into 2 sets, where one subset was used for the interaction modeling, and the interaction model was then used to calculate the PTOL and the NSRI and validate the prediction in the second population. And a PK of 1 means that there's a perfect correlation between the observed tolerance of laryngoscopy and the parameter which is investigated, and a PK of 0.5 is equal to flipping a coin. And we used bootstrapping to determine whether there was a significant difference in PK between the 2 parameters. So this table shows the PK values for each parameter. And as the 3 studies differ in which monitors were used, and the PK can only be compared between monitors if all the monitors were used in all the studies. So all studies used the bispectral index as an EEG monitor. Only the sevoflurane 2 sevoflurane studies used also the state entropy and response entropy as EEG monitors. And the sevoflurane-remifentanil interaction study also used the composite variability index and the surgical pleth index, which are both marketed as analgesic monitors and not so much as hypnotic monitors. And in all these comparisons, you find that the PTOL or the NSRI, which are this, of course, based on the same values, are significantly better predictors compared to any of the other parameters, with a PK of 0.92 and 0.95, whereas the other parameters are 0.78 or lower. And also, the effect site concentration of propofol is a very poor predictor and worse than any of the other predictors. And also, the surgical pleth index is a very poor predictor of tolerance of laryngoscopy. So an older version of the NSRI, which uses data from the 3 individual studies, is currently incorporated in the Smart Pilot View by Dreger. And also the Navigator by GE Healthcare incorporates several interaction models, with the exception of an interaction model between propofol and volatile anesthetics. And a small validation study by Cirillo et al. In 2015 was performed using only sevoflurane and remifentanil in 60 patients. And they found that the use of either monitor resulted in lower sevoflurane and tidal concentrations and higher BIS or entropy within what is considered the therapeutic range, without any differences in hemodynamic responses during surgery. And although this is not a validation of our triple interaction model because it wasn't used, it does show that a monitor that includes interaction modeling as a clinical decision tool may result in a more balanced anesthesia, which, along with the good prediction probability that we found in our initial validations, is promising for the use of NSRI in clinical practice. So in conclusion, we developed a 3-drug interaction model with an additive effect between sevoflurane and propofol and a synergistic effect between these 2 drugs and remifentanil. And this can then be used to improve on the already well-known concept of MAC and by transforming this interaction parameter into a promising clinical parameter for the tolerance of noxious stimuli in multi-drug anesthesia, the Noxious Stimulation Response Index. And the final model is relatively simple, and the first validation is promising. Of course, the NSRI still needs to be prospectively validated, but after which the possibilities are nearly limitless to be expanded with other drugs that can have an influence on tolerance of laryngoscopy, such as opioids, benzodiazepines, or alpha-2 agonists, such as clonidine or dexmedetomidine, or really any other drug you can think of that may interact with tolerance of noxious stimuli. So finally, I'd like to thank the team at the University Medical Center Groningen, Michel Struijs and Hugo Verreken, also in particular Hans Proost and Douglas Eleveld for their extensive modeling expertise. I'd also like to thank Martin Lugebul, Björn Heijsen and Thomas Bouillon for their valuable input. And of course, the original investigators of the 3 interaction studies in Ghent, Stanford and Bern, because without their hard work, there would be no triple interaction model. And thank you very much for your attention. Are there any questions?